Hi everyone! Today, in this video, we are going to explain the genetic basis of Down syndrome, leukemia, and its relationship. For this, we will first explain some basic concepts of genetics necessary to understand what we will explain later. Humans have 30 to 37 trillion cells, and these differ in about 250 different cell types that make up the different organs and tissues of our body. Cells have three main parts: the plasma membrane, that delimits the cell and controls what goes in and what's out, the cytoplasm, that contains the different organelles that perform a specific functions in the cell, and the nucleus, where the genetic material, or DNA, is located. DNA is the acronym for the biochemical substance deoxyribonucleic acid that contains the genetic information, that is, the instructions necessary for the development and proper functioning of the cells. It is similar to an instruction manual of the cells that contains the information to build necessary elements for cellular function. This instruction manual is written in 4 letters called nucleotides, which are joined in 2 chains, thus forming the DNA. They are made up of 3 parts: a sugar, or deoxyribose, a phosphate group, and a nitrogenous base. The nitrogenous bases of DNA are 4: adenine, guanine, cytosine, and thymine. These nucleotides are of complementary nature, so that A of one chain is always paired with T of the other, and the same happens with C and G. Thus, the nucleotides of each chain are joined by complementarity with the other chain to form the double helix of DNA. As we have said, these nucleotides would be the letters of the instruction manual, which are combined forming words that we know as genes. This information, through 2 complex processes called transcription and translation, translates into the specific combination of some of the 20 amino acids, so that specific proteins are formed, which have a specific function within the cell. Therefore, if the DNA information is badly written, changed, or there are segments in excess or missing segments, proteins may not be generated correctly and therefore cannot function, thus affecting the correct cellular function. Although we found the same genetic material in all the cells of our body, in each of them some genes are activated and some are not, allowing the cells to be different from each other, in order to perform the necessary functions in each tissue of the body, obtaining the cell types we have spoken before. But DNA is too long to fit inside the nucleus, and that's the reason that a group of proteins, named histones, join the double helix of DNA and enables it to compact, originating the chromosomes. The number of chromosomes is specific to each species. Therefore, in human cells we will find 46 chromosomes distributed in 23 pairs. One of these pairs determines the sex of the individual, and the other 22 are called autosomes. Although later we will continue explaining some basic concepts, we can begin to explain the issues that concern us. First, we will explain what Down syndrome is. Down syndrome is a genetic alteration present in 1 of every 700 newborns. People with Down syndrome have some distinctive characteristics, such as small heads and ears, short neck with a thickness in the back, flat skull, bulging tongue, ears tiled up, lack of muscle tone, and a unique palmar sheath. Babies with Down syndrome can be born with a size equal to the average, but they develop more slowly than children without said condition. In addition, they have more difficulty in achieving some abilities, and they suffer from having some degree of intellectual disability. It is necessary to emphasize that each case is different, since there is a great variability due to a very variable expression of these characteristics. But unfortunately, some medical complications can be associated with Down syndrome. In general, we can say that some of them have hearing or visual problems, a 40 to 50% have an associated heart disease, a 20 to 40% have hypothyroidism, a 2% leukemia, and other endocrine, neurological, or intestinal abnormalities, besides being quite prone to the development of infections due to some degree of immunodeficiency. But what is the genetic basis that originates it? Down syndrome is caused by the presence of an extra chromosome in P21. That is, people with Down syndrome have 3 chromosomes 21 instead of 2, so they have one more copy of that DNA segment. But how can they have one more chromosome? There are 3 main causes that can provoke trisomy 21. The first one is a meiotic mistake. The cells of our body are divided by a process called mitosis, so that out of a cell of 46 chromosomes, we will get 2 of 46. This happens in all the cells of our body, except in the reproductive organs, testes and ovaries, to obtain sex cells or gametes, oocytes and sperm. In this case, the division process is different. It is called meiosis, and in this, the genetic information is reduced by half. So at the end of the process, from one cell of 46 chromosomes, we will have 4 of 23 chromosomes, 1 of each pair. Thus, at the time of fertilization, a male gamete and a female gamete will match, forming the zygote that will have 46 chromosomes, just as their parents do. 95% of the cases of Down syndrome happen because of a mistake in the meiosis process, so that one of the gametes, usually the ovum, comes with an extra chromosome, which, when matched with the other gamete, originates a zygote with 47 chromosomes, and therefore Down syndrome. The second situation that we can see is the translocation. In this case, what happens is that a parent presents a balanced translocation, that is, he or she presents a fragment of chromosome 21 in another chromosome, usually the 14th. If at the time of meiosis both gametes contain the normal chromosomes, we will obtain a zygote with the correct number of chromosomes. But if a gamete contains a translocation of chromosome 14 with 21, the zygote will present 3 copies of a fragment of chromosome 21. It will not be necessary for chromosome 21 to be completely triple for these people to have the typical physical characteristics of trisomy 21, but this will depend on the translocated genetic fragment. And the third situation is mosaicism. In this case, the mistake occurs in mitosis, not in meiosis, after fertilization. If during the first meiotic divisions there is a mistake of separation in the mitosis process, all the cells that descend from the erroneous cell will present trisomy 21, but the rest will not. The physical features of the people with mosaicism and its potential development will depend on the percentage of trisomic cells that their organism presents, although they usually have a lower degree of intellectual disability. Once we have explained what Down syndrome is, let's focus on the second topic that interests us, leukemia. Leukemia is a type blood cell cancer. Cancer is an abnormal proliferation of a group of cells. It occurs when a cell accumulates damage in its DNA that is not correctly repaired, and instead of initiating an apoptosis process or cell death, it continues proliferation and accumulating mutations in an uncontrolled manner. Mutations are usually given in 3 types of genes. The first type are oncogenes. In their normal form, they are named proto-oncogenes. They control proliferation and cell differentiation and modulate cell survival. When they mutate, they become oncogenes, and alterations in proliferation and differentiation occur, as well as in apoptosis, favoring the uncontrolled growth of a group of cells. The second type of genes that can be mutated are thamossuppressor genes. Thamossuppressor genes have a wide variety of functions, very broad, many of them related to cell control, so they prevent this uncontrolled growth. If a mutation causes that they cannot perform their function, it will favor the appearance of cancer. And the third type of genes that can be mutated are DNA repair genes. They intervene in the repair of damaged DNA elements. Therefore, if they are not functional, the appearance of mutations will be favored. In the specific case of leukemia, this disorganized proliferation occurs in one of the blood cell subtypes. But how are blood cells formed? Through a process called hematopoiesis. Hematopoiesis occurs in some specific organs of the body, but at the beginning of human development, the fetus does not have hematopoietic organs. Therefore, the formation of blood cells occurs in the yolk sac until 12 weeks pregnancy. Although, from the 5th week, the liver already intervenes in the formation of blood cells, becoming the most important hematopoietic organ until 28 weeks. During this period, the spleen also intervenes in the hematopoiesis, although in a less way. As of week 35, the bone marrow is already configured as the main hematopoietic organ. Thus, during postnatal periods, the formation of blood cells or hematopoiesis occurs in the bone marrow, which is a spongy tissue found in sandstone bones, mainly in the tibia, femur, vertebrae and pelvis, sternum and ribs, even though once the growth is over, the long bones, that are tibia and femur, stop having hematopoietic activity. The different types of blood cells originate from the hematopoietic stem cells, located in the different hematopoietic organs. These have the typical characteristics of stem cells, which allow us to differentiate them from other cells. They are multipotent, that is, from them the different types of blood cells can be generated. They have a high proliferation capacity, and they have the capacity for self-generation, meaning the hematopoietic stem cells can generate hematopoietic stem cells. They are self-renewing. These stem cells, through the process of differentiation, originate the different cell lineages. Cell differentiation is a process in which genes expressed in the mother cell vary, thus expressing specific genes of a specific cell type. We can distinguish between myeloid stem cells and lymphoid stem cells. The first give rise to red blood cells, platelets, and granulocytes that are masts, basophils, eosinophils, neutrophils, dendritic cells, and macrophages, while the latter originate B lymphocytes, T lymphocytes, and natural killer cells. So we have three main types of blood cells: the red blood cells or erythrocytes, the ones that carry oxygen to the whole body; the platelets, which are involved in the start of clot formation to stop the hemorrhages; and white globules, formed by the cells of the lymphoid line together with the granulocytes, which fight the infections of the body. Leukemia is caused by the appearance of certain mutations in these cells' DNA that induce the uncontrolled proliferation of these mutated cells, invading the bone marrow and preventing the formation of normal cells. There are four main types of leukemia that differ in whether the cancer cells belong to the lymphoid line or the myeloid line, and whether they are acute, that is, if they affect less differentiated cells and therefore are fast-growing and sudden onset, or chronicals, that is, they affect cells in a more advanced stage of maturation, and therefore they grow in a more post fashion. So we can distinguish among acute lymphoblastic leukemia, or ALL, acute myeloid leukemia, or AML, chronic lymphocytic leukemia, or CLL, and acute myeloid leukemia, or CML, and within them there are several subtypes. Once we have explained Down syndrome on the one hand and leukemia on the other, we will see the relationship that unites them. Children with Down syndrome are 20 to 500 times more likely to develop acute lymphoblastic leukemia and acute myeloid leukemia respectively, compared to children without Down syndrome, although they are less prone to develop solid tumors. Although acute lymphoblastic leukemia is the most common form of leukemia in children with and without Down syndrome, today we are going to talk about TMD and AMKL, which usually only occur in children with Down syndrome. Approximately 10 to 15% of children born with Down syndrome develop during the first 3 to 7 days of life transient myeloproliferative disorder, which is a pre-leukemia characterized by an accumulation of immature megakaryoblast in the fetal liver, peripheral blood, and bone marrow, and a decrease in the number of platelets and erythrocytes in blood circulation, although of the latter in a smaller proportion. Most cases tend to revert without treatment within 3 months, but unfortunately, 20 to 23% of survivors end up developing AMKL before the first 5 years of life. AMKL, or acute megakardioblastic leukemia, is a subtype of acute myeloid leukemia, specifically the M7-AML subtype, and 99% of cases occur in children with Down syndrome, and is characterized by an uncontrolled proliferation of malignant megakardioblasts that affects various tissues. TMD is caused by a cooperation between trisomy 21, which alone disturbs fetal hematopoiesis, and mutations acquired in a gene called GATA1. Both elements are enough and necessary for the development of TMD, since mutations in GATA1 in the absence of trisomy 21 have not been associated with the development of leukemia. Abnormalities in hematopoiesis due to trisomy 21 begin in the embryonic yolk sac, and both in this hematopoiesis and the one in the fetal liver, there is a shift towards the megakaryocytic erythrocyte production line. In some experiments, it has been observed that hematopoietic stem cells and myeloid progenitors with trisomy 21 proliferate more than normal cells. So, it is believed that trisomy 21 alone causes an increase in the number of megakaryocyte erythrocyte progenitors, or MEP. And it is also believed that common myeloid progenitors are also altered, favoring erythromegakaryocytic production to the detriment of granulocytic. Despite not being entirely clear, it is believed that this alteration in hematopoiesis is due to small changes in expression of multiple genes found on chromosome 21. It contains several genes involved in hematopoiesis, such as ERG, RUNX1, ATS2, SON, and DYRK1A, and some investigators tend to think that more than an overexpression on one of them, there is a change in several of them, causing this hematopoietic anomaly, although some scientists believe that some have an effect greater than the others. It is still under study. In addition, trisomy 21 causes extensive changes in the genome and gene expression, affecting multiple genes in multiple chromosomes. As we have said, the second element necessary for the development of TMD is the presence of mutations in the GATA1 gene. Thanks to studies of new generation sequencing, it has been seen that all the cases of TMD and AMKL present mutations in this gene. It is located in the X chromosome and codifies for two proteins, GATA1 and GATA1S. GATA1 is a transcription factor, that is, a protein that binds to DNA regulating the expression of other genes. In the specific case of GATA1, it is a key transcription factor in the differentiation of erythrocytes and megakaryocytes. Mutations in the GATA1 gene provokes that only the protein GATA1S is inside. It is a truncated protein. In other words, it's a protein with a part deleted, and therefore cannot interact as it should with DNA and other proteins. So, it is less effective regulating the differentiation of erythrocytes and platelets, and instead favors the proliferation of megakaryoblast. These mutations occur during fetal development, since they can be detected in fetuses at 21 weeks. These two alterations are enough and necessary for the development of TMD, and in most cases, there is a reversal of the disease in the absence of treatment during the first 3 months of life. But before, we said that a percentage of these children end up developing AMKL before age 5. This is because in some cases, all the blasts that show altered trisomy 21 and GATA1 are not completely removed, and in this appear more mutations in other genes of other chromosomes that provoke the development of this leukemia. In addition to this, studies with new generation sequencing techniques have shown that 30% of children with Down syndrome have mutations in GATA1, although only a 50% develop TMD. These cases are known as silent TMD, but it's very important to take them into account since these children are also susceptible to developing AMKL. Children with AMKL need chemotherapy treatment, but in general they usually respond quite well, presenting a high survival rate. In summary, we can say that the presence of trisomy 21 in hematopoietic progenitors causes an alteration of the hematopoiesis. In 30% of those born with Down syndrome, mutations in the GATA1 gene appear during fetal development, although only 15% have TMD. Most reverse spontaneously, although among the survivors, a 20 to 23% of cases develop AMKL due to the appearance of mutations in other genes. And this is the information we consider necessary to understand the genetic basis of leukemia in Down syndrome. If you want to go deeper into the subject, you can consult the bibliography used to make this video that is at the end of it. Thanks for your attention. I hope you have understood all the information given in this video and that it has been helpful. Have a nice day.